as we left off, we were talking about the steps of glucose oxidation in various steps. And the first step is going to be glycolysis. And glycolysis does not use oxygen. As we will see, it uses substrate level phosphorylation to phosphorylate ATP. Then a pyruvate, which is the end product of glycolysis. Pyruvate is the end product of the glycolytic pathway. Pyruvate is then oxidized, and then the product of that enters a cycle called the Krebs cycle, also known as the citric acid cycle. And then finally, the products of the Krebs cycle and a few products of glycolysis, they come together, and those products enter an electron transport chain. And there is a process called chemiosmosis, which we shall cover in great detail. But right now, let's focus on glycolysis, process of glycolysis. That's what we're going to tackle. But before we do, let's look at where all this takes place. Glycolysis, in which glucose produces two molecules of pyruvate, as we will see, with some yield of ATP and also a yield of NADH, that is reduced NAD. Glycolysis takes place in the cytoplasm. But pyruvate oxidation, the Krebs cycle, and the electron transport chain involving chemiosmosis to produce ATP all occur in the mitochondrion. So we'll need to consider the inner membrane of the mitochondrion and the matrix of the mitochondrion where this takes place. But right now we're concerned with cytosolic process, the cytosolic process of glycolysis. As I've said, glycolysis converts glucose to pyruvate in a 10-step biochemical pathway. We've talked about what biochemical pathways are, so you should be familiar with that. This occurs in the cytoplasm, as I've mentioned, and there are two molecules of pyruvate formed for every molecule of glucose that enters the glycolytic pathway. Two molecules of pyruvate are formed. And there's a net production of 2 ATP by substrate level phosphorylation, as I've mentioned, and 2 NADHs are produced by reducing 2 NADs. And we'll see this all very shortly here. So let's look at first an overview of glycolysis. We're dealing with glycolysis here. We start with a 6-carbon glucose molecule in which we have to input energy into the process at the beginning. So there is an energy investment phase at the first part of glycolysis. And we actually use ATP, donate phosphates from ATP, yielding ADP, to phosphorylate a 6-carbon sugar. We start with glucose, which is 6-carbon sugar. We convert glucose, as you'll see in a couple of steps, to other 6-carbon sugars, and that is phosphorylated twice, as we will see. So ATP is invested, in a way. We're putting energy into the system at the beginning. We're priming the reactions. So you're saying, wait, I thought glycolysis in it and the burning of foodstuffs yields ATP. Why is it that we're investing ATP? Well, we need to invest ATP at the beginning to prime the process. And then there is a cleavage stage of glycolysis in which the 6-carbon diphosphate molecule is split into 2 3-carbon phosphorylated molecules. And then each of those is run through the same pathway. So we can ignore one side, really. Let's just look at one side. Just know that this happens twice because there are 2 3-carbon molecules produced in the cleavage phase. And then as those are run through the last phase of glycolysis, this is where oxidation occurs and NAD plus gets reduced to NADH. We oxidize compounds here. And in the process, we also yield ATP here. And we yield 4 ATPs where we only invest 2 at the beginning. So we have a net yield of glycolysis of 2 ATPs. So energy investment, cleavage, and then energy yield, producing NADH and ATP. All right, we left off with the net yield of 2 ATP in the overall process of glycolysis. Now let's look at this biochemical pathway in a little more detail. So we're going to start out with glucose, and in step 1, the enzyme hexokinase phosphorylates at substrate level phosphorylation, phosphorylates glucose to yield glucose 6-phosphate. Here we have glucose, and here we have phosphate attached to the 6th carbon of glucose. There's an oxygen and then 1, 2, 3, 4, 5, 6 carbons. Glucose is a hexameric carbon sugar. So hexokinase takes a phosphate from ATP and attaches it to glucose to yield glucose 6-phosphate. You might ask, well, what is the delta G for this reaction? So let's look at that. Delta G0, and by delta G0 we mean standard conditions, maybe not the conditions that are existing in the cell, but if we were in a test tube with standard concentrations of reactants and at standard temperature, we have a delta G of -4. That's negative, so that's a spontaneous reaction. Why is that spontaneous? We have a negative delta G because ATP, we are hydrolyzing a high energy phosphate bond, getting rid of one of the phosphates, one of the negatively charged phosphates from ATP, which yields energy and allows this reaction to proceed forward with a negative delta G. So we have glucose 6-phosphate, and in step 2, an isomerase comes in. You know what isomers are. They are the same molecular formula, but different arrangements of the atoms. And so phosphoglucose isomerizes glucose 6-phosphate to fructose 6-phosphate in step 2. And here is fructose 6-phosphate. This is a complex process, but the net result is an isomer of glucose 6-phosphate, fructose 6-phosphate. What is the delta G there? Oh, it's a little bit positive, but very close to 0. So it's a net, there's not much energy that needs to be transformed here. So this proceeds, assuming there's glucose 6-phosphate around, this catalysis will proceed under the enzymatic activity of the isomerase. Now, in the third step, fructose 6-phosphate here is phosphorylated with another substrate level phosphorylation event. So this is phosphofructokinase. Kinases are enzymes that phosphorylate compounds. So here a phosphate is donated from ATP, and now we have, at the end of this phosphorylation, we have fructose 1,6 bisphosphate or biphosphate. So now we have fructose 6-phosphate, but now the first carbon also has a phosphate on it. So we have 2 phosphates on fructose 1,6 bisphosphate. And phosphofructokinase is actually a regulatory step. This can be allosterically regulated by negative feedback inhibition. We're not really going to go into that, but this is a regulation step for glycolysis, whether or not glycolysis will proceed. Phosphofructokinase can be inhibited by allosteric inhibition. And you should know what that is by now, I hope. So we have, at the end of step 3, we have fructose 1,6 biphosphate. The delta G there was negative 3.4. That's to be expected because we are hydrolyzing a high energy phosphate bond in the donation of a phosphate group from ATP, yielding an ADP molecule. So you can see we've already invested 2 ATPs here and yielding 2 ADPs in this phase. Now, the next step is there's really a 2-step reaction. Aldolase is the enzyme, and then isomerase also is the enzyme. They act in concert here. And let's first look at aldolase. Aldolase breaks the 6-carbon fructose 1,6 biphosphate molecule into 2 3-carbon molecules. One is dihydroxyacetone phosphate, and the other one is glyceraldehyde 3-phosphate. So here's glyceraldehyde 3-phosphate over here, and here is dihydroxyacetone phosphate. But then the isomerase plays a role and converts the dihydroxyacetone phosphate here into glyceraldehyde 3-phosphate. So we end up with, at the end of steps 4 and 5, we end up with 2 molecules of glyceraldehyde 3-phosphate, which each of which enters the next stage of the glycolytic pathway. So we've gone through the energy investment phase up here, and then the cleavage phase by aldolase, and then the isomerization of dihydroxyacetone phosphate into G3P, glyceraldehyde 3-phosphate. And now we are ready to enter in stages 6 through 10, we're able to enter the energy yielding phase. So the delta G's for these reactions are interesting. Look at the delta G for the cleavage, the cleavage of fructose 1,6-bisphosphate into these two 3-carbon molecules. It's very positive. It's plus 5.7, and that deserves a comment, which I'll get to in just a second. So we have a very high delta G0 here of plus 5.7, and you must be thinking, how can this reaction possibly proceed when this indicates non-spontaneity? How is it that the relevant enzyme catalyzes this cleavage of fructose 1,6-biphosphate into these two 3-carbon molecules with a delta G so high, so positive? And here I want to introduce you to the concept that although this is under standard conditions, in the cell, the delta G can actually be 0. Why? Because fructose 1,6-biphosphate is rapidly converted in the above reactions to, is rapidly produced rather by the energy investment phase. And so there's a very high concentration of fructose 1,6-biphosphate. And on the other hand, glyceraldehyde 3-phosphate, which is the end product of these coupled reactions here, 2 molecules of glyceraldehyde 3-phosphate, which are going to enter the next phase, these are rapidly are utilized in the downstream energy utilization phase. So the concentrations of product here are very low because they are rapidly utilized. And the concentration of reactant is very high because of it being produced very rapidly by the energy investment phase. So my point here is that the high concentration of reactant and low concentration of product can cause the pushing of the reaction because of high concentration of reactant, and then the sucking, the pulling, I'll say, of the glyceraldehyde 3-phosphate product down the pathway. So in actuality in cells, this delta G is not delta G0, but delta G is actually negative. So this introduces you to the concept of pushing biochemical reactions and pulling them by the relative concentrations of reactants and products. In this case, the products are rapidly utilized and the reactants are produced at high concentration. And so we push this reaction from above and pull it from below. You could think of a car that's being pushed. You're pushing the car, but you're also pulling the car with a tow truck or something. That allows for this reaction to proceed. And although you'll see at several steps in the glycolytic pathway, there are positive delta G zeros, the overall delta G is negative for the entire pathway, and that causes the pushing and pulling of different reactants and products through the pathway. So in our treatment of glycolysis, we've gone through the energy investment phase here, and we've gone through the cleavage phase. And now it's time to enter the energy yielding phase as 2 molecules of glyceraldehyde 3-phosphate, which are the end product of the above 2 phases, enter the energy yielding phase. So we're entering the energy yielding phase now, and that's where we'll pick up within the next segment of this lecture.